Now we need to move to a new tool called cell detection tool. And the cell detection tool automatically identifies and segments individual cells in histopathology images, distinguishing between nuclei and cytoplasm when possible. It uses intensity, color, and morphological features to detect cells accurately, even in densely packed tissues. This tool is essential for quantifying cell populations, assessing biomarker expression, and supporting diagnostic or research workflows, providing reproducible high-throughput analysis while allowing manual review and correction to ensure the precision. So simply, the cell detection tool automatically identifies and segments individual cells in tissues for the different quantitative analysis. To access this tool, go to Analyze and then Cell Detection and then select Cell Detection. If you applied cell detection right now and pressed on Run, it will work on the whole image or the whole slide image and start to find the cells inside it. And it will be time consuming depending on the shape or the size of the whole slide image. Another approach is to select a specific annotation or a specific rectangle or a polygon and inform that cell detection to work on the internal region of that annotation. So simply, let's go to annotation and let's say we need a region, press on auto set, and let's say we are selecting that region. Once you are selecting that region and press on run, it will only calculate and define and identify the cells inside that region. You can now look closer to each nuclei as you see here. So instead of manually segmenting all of them, you can simply run the cell detection to detect all of the cells inside that region. You can get the meaning of these different options from the manual, or simply you can put your arrow on any of these options and it will show a tip of the meaning behind that option. For example, the cell expansion mentions there is the amount by which to expand detected nuclei to approximate the full cell area. And simply this means there is expansion outside the nuclei. And as you see here in the figure, there is two contours, one for the nuclei and another for the expansion. So if we decrease that, let's say, to 1 micrometer and press run again, it will decrease expansion. So let's zoom in. So the expansion here is only 1 micrometer outside the boundary of the nuclei. You can ignore it also by making it 0. Run again. As you see here, it only detected the nuclei. And this option can be useful as the cell detection tool is not accurate 100%. It may detect only a specific portion of the nuclei, such as here, where this portion should be also included. So when we do cell expansion by a specific distance, then we can get the outer polygon instead of the nuclei polygon to be in the safe side and to be sure that it includes the whole nuclei. Also, you can change the threshold to filter out some nuclei that doesn't exceed the threshold. So for example, if we set the threshold by 0.5 and zoom out like that and press run, some of the nuclei, as you see, are ignored, and only the nuclei that exceeds or equal to that threshold will be detected. And this simply means that the nuclei with lower intensity values or has a lower mean intensity values will be ignored because it doesn't exceed or equal to that threshold. Let's return it to 0.25. And there are other two options, which is the minimum area and the maximum area. And here we are setting constraints on the size of that nuclei. So if the area below the minimum, it will be ignored. And if the area is exceeding the maximum, it will be ignored. So let's say that we need a minimum area of 50 and a maximum area of 100. And press run again. So as you see here, this nuclei and this one and this one, and just few of the nuclei inside the annotation that satisfy those conditions with a minimum, maximum, and the defined threshold. Let's return it to 10 and keep this one to 50 and run again. For sure, the number of the nuclei which is detected by this tool will change according to the new criteria. And as you see here, if you zoom in, this nuclei has an intensity that exceeds the threshold and within the specified area. Also, you can make measurements here so that later you can get some features from each nuclei, as we are going to see. But for now, if you go to hierarchy, the number of detected nuclei is around 22K. So here, using this tool, you are adjusting the configurations according to the problem and the dataset to keep only the nuclei that satisfy specific conditions, and later you can use these nuclei features in your machine learning pipeline. By getting assured that these configurations are working and satisfy the requirements of your project, you can create another workflow as we did with the stem vector estimation. So go to workflow and select cell detection. Ensure that these are the configurations that you are selecting, and then create a script. This command is the last one. So go to the last command, which is this one. Copy that command and remove others. Just keep the last command here, and then save that as a Groovy script. Let's say cell detection. Now we have a script that can be run and expanded to other whole slide images. Here we are in slide 5. So let's go to slide 4, save, go to this region. Let's create a rectangle like that, and then go to automate, script editor. You have the cell detection here. Select that and run. It will run on that specified region and detect the cells within the specified constraints that you put here and configured in slide 5. And as you see here, only a set of nuclei that satisfy the conditions are selected and others are ignored. Now let's move to another tool called positive cell detection tool. And this tool builds on the standard cell detection by identifying cells that express a specific marker, and it classifies cells as positive or negative based on user-defined intensity thresholds, enabling quantitative assessment of biomarker expression within tissue samples. And this is typically valuable in research and clinical contexts, such as evaluating immune cell infiltration or hormone receptor status, where accurate counting of positive cells directly impacts the interpretation and decision making. So simply, the positive cell detection tool automatically identifies and counts cells expressing a specific marker based on user-defined intensity thresholds. Now, to access the positive cell detection tool, let's go to Analyze, Cell Detection, and Positive Cell Detection. And here you will find similar options as cell detection, but in positive cell detection, you will get intensity threshold parameters, where here the tool is distinguishing between positive and negative. Threshold 1 is the low positive intensity threshold, 2 is the moderate one, and 3 is the high one. By default, if you run this tool, it will be executed on the whole slide image, but here we need only a specific portion to apply the tool on. We selected this region, and let's keep these parameters as is and run the tool. As you see, the tool detected many positive cells in red and a set of annotations here which are negative, and both detections are depending on the thresholds that you are defining here. Let's cancel the cell expansion, run again. If you zoom in, you will find here positive and negative, positive in red, negative in blue. You can also change the threshold, let's say 0.25, run. As you see here, the cells that has a mean intensity value below the threshold will be ignored. Now we have here negative, negative, positive, positive, and so on. You can also define the minimum and maximum area Let's keep it for now by default 10 and 400. And this option, which is single threshold, it classifies the nuclei or the detected nuclei whether it's positive or negative. But if you need the grades of it, depending on the thresholds here, you can unmark this option and press run. It will classify each of them depending on the threshold, whether it's low positive, high positive, or moderate. And you can find that in the hierarchy under this annotation. We have negative cells here. This detection 1 plus, this means it's low positive intensity threshold. You can also find 2 plus as an example here. And we may also find 3 plus, which is high positive intensity threshold. So depending on the thresholds and the mean intensity of each nuclei, it will be classified accordingly. So again, using this option, you will classify them into pure positive or pure negative. If you didn't select this option, it will be negative, and other three classes, +1, +2, +3, reflecting low, moderate, and high positive intensities. Also for this tool, you can create a script. So go to this last one and ensure these are the configurations that you are targeting. Create the script, take the last script that you executed and remove other ones. Save the script. Let's call it positive cell detection. Save the script, and you can go to any other annotation. Let's say this region. Let's create an annotation like that. Go to automate, script editor, select the positive cell detection and run. It will run on that region. You can also apply that in another whole slide image. As you see, there is negative, there is +1, +2, and the red one is +3. If you go to the hierarchy, open the annotation, is an example of +1, which is in yellow. This is another example of +2, which is in orange. And there's an example of +3, which is the high threshold. Now let's move to another tool called fast cell counts, and it provides a rapid automated method to estimate the number of cells, typically the nuclei, in a selected regions without performing full segmentation or detailed morphological analysis. This tool is optimized for speed and efficiency, making it ideal for quick assessments or large datasets where high precision is less critical than throughput. While less detailed than the standard cell detection tool, this tool offers a practical balance between the performance and accuracy for preliminary screening or resource-limited scenarios. So in short, this tool quickly estimates the cell numbers in a region without full segmentation, which is ideal for rapid screening. To access this tool, go to Analyze, Cell Detection, and then Fast Cell Counts. Again, create a region like that or annotation and keep the parameters like that by default and press run. It will detect the cells and count them faster than the cell detection tool. And you can change the parameters here. Let's say the cell detection threshold is 0.5. So it counts only the number of nuclei that exceeds that threshold. As you see here, you can change the diameter to 5 so that it decreases the diameter of the circle, or keep it, for example, 10, like that. So we have cells here like that. You can change also the sigma, and this is a value used for the smoothing, which is a filter applied so that it can detect the peaks in the intensity and therefore detect the nuclei based on that. To get the count of the detections, we have now 16 cells that satisfy this criteria. And you can also go to hierarchy or the annotations, and inside this region we have different detections. If you click on any of them, you will get the centroid X and centroid Y. And notice that there's only for detection. But for cell detection tool, you will get more properties alongside the centroids, such as area, perimeter, circularity, and so on. So you have different features which are calculated from the cell detection tool, and inside the fast cell counts, you only get the centroid X and centroid Y. And that's why the fast cell counts is considered faster than the cell detection tool. However, this tool doesn't perform any morphological analysis to calculate any features. So if you are targeting a way to count the cells inside an annotation or a region without considering the features, then you can use the fast cell counts. If you need a tool that can gather the different morphological features, then use the cell detection tool. If you need a tool that can classify between these nuclei or those cells, use the positive cell detection tool. And again, for the fast cell counts, you can create a workflow based on these configurations. Now, let's move to another tool called Pixel Classifier tool. And this tool enables users to train a machine learning model to classify individual pixels in an image into user-defined categories such as tumor, stroma, background, based on color, texture, and intensity features. By manually labeling representative regions, the tool learns to generalize these classifications across the entire slide, facilitating automated tissue segmentation and quantitative analysis. This is particularly useful for complex and heterogeneous samples where rule-based methods may fall short. So simply, this tool uses machine learning to automatically label pixels in an image based on user-defined tissue categories. Let's go to annotations and delete all of the annotations. It may ask you if you want to keep the descendant objects, which are the inner nuclei or regions. So press on no. All of the regions are removed. Now you need to manually label some regions in each category. So let's say that I'm selecting now positive, auto set, and I will select the one tool here. Let's say this is positive, positive here, 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 and here, here. And the more you label, the better you teach the classifier. Now we have 10 objects or annotations for the positive. Now let's go to negative. Let's say this region, this region, this one, this one, this one, here, 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 and here. Now we have 10 annotations in the positive and 10 annotations in the negative. Go to classify, then pixel classification, and select train pixel classifier. You can toggle whether to calculate the classification live during viewing the image or not. Now, as you see, it started to classify all of the pixels, whether it's negative or positive, like that. And as you see, it may not be accurate 100% because the number of annotations are not too enough for the whole slide image. So it's better to get more annotations. Let's say we need another class here, path, and annotate this region, here, here, and then let's go to this region like that, and exclude the inner portion by pressing on Alt Shift, as we mentioned, to exclude a specific region like that. Let's go here, annotate this region, this region, here, and here. Also, these are also can be categorized as fats. And for positive, let's say this region, here, 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 like that. And for negative, let's say this one, this one, this one, and here. Now let's try again to classify. Train pixel classifier, turn on the live prediction. It started to learn and predict using live prediction. So simply here tries to learn from your annotations to classify the pixels based on these annotations. So the more data you are giving to the classifier, the better the classifier learns and classifies. For those options, you can select different classifiers. So let's keep it by default, artificial neural network, and you can increase the resolution. So the full resolution is the highest one. If you press on it, it will increase the resolution of the classification. Also, you can edit the features by going to edit and select which channels are you working on. So here we are working on hematoxylin and uosine, so ignore those channels and press OK. Now it repeated the training and the classification, and here it shows that most of the regions are fed because we didn't classify what are those. So let's say that to be ignored. So go to ignore and select a big portion here using the one tool like that, something like that. And here, without selecting any nuclei, go here, the same, try to select a reasonable portion. Now we have 4 categories. Go to classify, pixel classifier, claim pixel classifier. Ensure that the features are eosine and hematoxylin. Ignore these 2 features, press OK. Live prediction. Now it ignored these regions and only classified negative, positive, and fat. So this region is considered fat, it's classified. These regions are nuclei, are selected to be positive. These regions are selected to be negative, and so on. So the more data and labels you are defining to that classifier, the more precision and accurate the model becomes as it learns from your data. So once you are satisfied with that classification, you can save it later for other classifications by simply setting a name for it. Let's say 4 categories like that, which is fat, ignore, negative, and positive, and then save it. So it's saved, and you can load it later to be used to classify other regions or other whole slide images by simply going to classify, pixel classification, load pixel classifier. Now we have for CAD, apply the classification to be like that. You also can go to another slide and load that classifier, and it will start to classify the regions based on the trained data from the previous slide. So simply to build that classifier, you need to correctly annotate a set of regions depending on the classes you are selecting. Let's say positive, negative, ignore, and any other classes. Do that for a reasonable amount of annotations. Create the classifier and configure it and save it for later usage. Load it in any other whole slide images, and you can classify based on the trained data. Also, if you clicked on create objects and select the full image, you can create objects from that image under annotation or detection object type and configure the minimum object size, the minimum whole size, these options. Let's say spread objects and press OK. As you see, it started working, and after completion, you will find that you have around 38 positive, 36 as fat, and these are the different objects. You can select any of those and refine them. Let's say I will select, for example, this positive, which is here, this one, which is here, and so on. So it's very simple. You don't have to annotate the whole slide image. Just train a classifier, and it will do the process on your behalf. Let's return back to slide 4, and we need to store those measurements as a file so that we can use the features later in any programming language. So simply go to measure and export measurements. You can select which whole slide image do you want to work on. Let's say I want to work on 4 and 5. Here, define the output file. Let's say measurements, and select the separator to be comma CSV file, and which export type you are targeting. Let's say annotations, and export. It will export into this file. You can open it using any CSV reader like Excel. Open with Excel. Now you have the image type 4.svs and 5.svs, and depending on the number of annotations, you will find that CSV file is populated with all of the annotations. And here you are getting the different features like centroid, area, and the perimeter. Also, you can export the different detections. Let's say I need all of the detections in all of the images and store it as CSV file, and let's say measurements2.csv, and export that. Open it again using Excel for measurements2.csv. You will find more features because the annotation features are just centroid, area, and perimeter, but the detection, which is a result from positive cell detection or cell detection, has more features such as hematoxylin OD range, eccentricity, circularity, and area. Finally, you can export the annotations as a JSON file so that you can use the coordinates of these regions later with Python to extract them using a programming language. One of the options, you can go to File, Export Objects as GeoJSON, and you can select whether to export all objects or selected objects. Let's say all objects, and don't exclude the measurements, and we don't need any compression. You can select pretty.json so that you can make the file readable when you open it. Press on OK, export as GeoJSON file. When you open it, you will find the type, which is polygon, and these are the coordinates for the first one, and these are the properties for the polygon, and the same for others. You can also find more tutorials and information about the different tools and other tools that we didn't discuss in this session in the QuPath documentation. Simply go to qpath.github.io and scroll down and find the documentation. You will find different tutorials here regarding the different options of the QuPath. For example, for cell detection, it shows a tutorial on how to use the cell detection, positive cell detection, and so on. So, in this lecture, we have covered a set of tools for QuPath and mentioned that QuPath is a powerful open source software that bridges digital pathology and computational analysis. We covered different annotations such as wand and brush, advanced tools, and other simple tools like the polygon and rectangle. We went through how to select, deselect, and change the opacity. We knew how to estimate the vectors and moved after that to cell detection using cell detection, positive cell detection, and fast cell counts. After that, we discussed how to use the pixel classifier, train it and load it later in other slides and annotations to be classified. Finally, we mentioned how to export the annotations and results, whether using the export measurements tool or by using GeoJSON export tool. In the end, we covered how to navigate the documentation to gain more tutorials or to read more about the different concepts or how to use any of the discussed tools.